Assalamu alaikum everyone. Today we are discussing about PET scan or the PET scan. It stands for positron emission tomography. It is actually a technique that is used to reveal the biochemical functioning of our underlying body structures like tissues and organs. Or we can also define it as a technique that is used to identify how well our body organs are functioning. It is a very important diagnostic tool in conditions like cancers and tumors. But the question remains that we already had a large number of imaging modalities, like there was MRI already available, there was CT scan already available. So what actually was the need of coming up with a technique as that of a PET scan? Basically, the CT scans and the MRIs are actually the structural imaging techniques, while as the PET scan is a functional imaging technique, which means the CT scans and the MRIs were helpful in revealing the structures of your underlying organs and tissues, while as the PET scan is helpful in revealing the function of your body organs. The key player of PET scan is a subatomic particle called as positron, which is basically a counter particle of electron. That means that electrons and positrons are both subatomic particles having similar mass, similar charge, except for the fact that positrons have got a positive charge, whereas the electrons have got a negative charge on them. And there's a very important concept in physics. We call that concept as annihilation. As per physics, annihilation is a reaction where a particle collides with its antiparticle and disappears, liberating a large amount of energy in the form of gamma radiations. This annihilation basically forms the basic principle, working principle of our PET scan. By the end of this video, we'll be able to understand why the PET scanning is done, how is it carried out, what's the structure of PET scan, what is the biochemistry behind the PET scanning, and how can we interpret the results of a PET scan. PET scanning consists of a machine called as PET scan machine, which is kept in a radiation-proof room. It consists of a tunnel to allow the entry and exit of the patient, and it also has a scanner in it to identify some radiations. We'll be discussing them later. There is also a computer that is kept in an adjacent room in order to operate the PET scan machine. Now, we already know that for our body cells to function and to survive, they need a lot of energy in the form of ATP. And this energy comes from the glucose that is present in our blood. Let's suppose these are the healthy cells of our body, and this is the blood vessel that carries blood in it. Blood contains dissolved glucose. Every time these cells require energy, they will pick up glucose from the blood, break it down and release energy in the form of ATP. This is the basic principle that was exploited by scientists, and they developed an analog of glucose. We call that analog as 18-FDG. FDG stands for fluorodeoxyglucose. It is basically a glucose analog which, when consumed by the cells, releases positrons. And once the positrons are released inside the cells, the positrons react with the electrons that are already present inside the cells. They annihilate and liberate a large amount of radiation in the form of gamma radiations. These radiations are picked up by the scanner. What we basically do is, the patient whose PET scan is to be performed, he is given an intravenous administration of this 18-FDG. This 18-FDG enters the blood of the patient. Now what happens is, these cells will now uptake 18-FDG instead of normal glucose. 18-FDG will enter the cells, it will be broken down, and upon its consumption, it will release positron. Positron again will react with the electron, annihilate and release gamma radiations. Now, there are basically two situations that may arise. Let's consider situation number one. This is, let's say, an example of the healthy cells. The healthy cells show a normal activity. There is a normal metabolic functioning of the healthy cells, and they will uptake normal, or we can say less quantity of the 18-FDG. Less and less FDG will be taken up, which means less and less positrons will be emitted, which means less and less annihilation will occur, and as a result of the radiation that will be emitted, they would be of low density areas. We call these low density radiation areas as the cold spots. Situation number 2, let's suppose we have the cancerous cells in our body. We already know that the cancerous cells are the glucose hungry cells. Cancer is basically an unorganized mass of the cells that has a capacity to infect other healthy body cells. These cancerous cells show a hyperactivity, and as a result of this, they would demand more and more FDG from the blood. They will uptake excessive FDG from the blood, and as a result, excessive positrons will be emitted, and consequently, excessive radiations would be emitted. Those areas where the radiations have a very, very high density, we call these areas as hotspots. Now, once we get the PET scan of a patient, be it a healthy or an infected patient, if the PET scan shows 2 areas, the low density areas and the high density areas, that is the hot spots and the cold spots, we can infer from that the patient might be suffering from conditions like tumor and cancer. This was all about PET scanning. Thank you so much for your time.